Okay, so while there are presynaptic receptors, both autoreceptors and heteroreceptors, where drugs may act and where neurotransmitters may act, remember the autoreceptors are presynaptic and they're going to bind the neurotransmitter that is released by the same neuron that's expressing them. So if this is a dopaminergic neuron, we'll have a dopamine receiving, right, dopamine binding autoreceptor, and that will largely often, anyway, serve sort of a thermostatic role, sort of reduce, the further release, of that dopamine. Or we could have a heteroreceptor. This is another presynaptic receptor, right? This is something that will bind a neurotransmitter that's released from another neuron in the vicinity, like let's say acetylcholine. If this is a dopaminergic neuron and it has a receptor that's going to bind acetylcholine, well, that cholinergic receptor, the receptor binding the acetylcholine, is a heteroreceptor because it's binding something different than the neurotransmitter that this neuron releases. But the majority of the receptors we're going to find are going to be found post-synaptically, so on the next neuron, on the dendrite, let's say, the next neuron. And there are going to be a number of different types of receptors that we can find. In fact, for just one neurotransmitter, let's say serotonin. Serotonin is a very complex kind of neurotransmitter system where you have, about 14 distinct serotonergic, we're going to say serotonin binding or receiving receptors that are, you could potentially encounter in the postsynaptic membrane. So it gets very complicated, right? So what we can do, though, is sort of divide up the different types of receptors, the many different types of receptors, but put them into, fewer sort of classes based upon certain structural and functional responses that they generate in response to the binding of neurotransmitter. So one of the things to consider or to keep in mind about all receptor proteins, they're embedded in membranes, right? The ones that we're talking about are going to be embedded in the presynaptic or the postsynaptic membrane of the neuron. They receive, the chemical, like the neurotransmitter that binds to it, or a drug can bind to it, right? And when a chemical, whether it's a neurotransmitter or a drug, binds to a receptor, whether it's an autoreceptor or heteroreceptor on the presynaptic membrane, or whether it's one of these postsynaptic receptors, the binding changes the shape, the structure of that complex, usually transiently. Some drugs bind sort of more permanently, and that can be very dangerous, right, depending on the drug. But generally with neurotransmitters, it binds to its receptor. There's a change in shape, which changes the function. It does something new, and then it gets released. And it goes back to its shape. So there's a kind of a temporal, aspect to this of, the binding, the change in shape, and then a functional change because there's been a structural change, and release and then return. And what you're seeing here on this video is, or this slide, is the neurotransmitter being released. Remember, it's being released from the vesicles upon the arrival of an action potential, the opening of the voltage-gated calcium channels, the fusion of the vesicles with the presynaptic membrane. You had all that stored up concentration gradient force because of the vesicular transporter, using ATP to pack those vesicles in advance with neurotransmitter. And now it's going to diffuse out. It's going to work its way out, based on the concentration gradient force. So some percentage of it, is going to there's some probability that some percentage will actually reach and bind with these receptors that are located on the postsynaptic membrane. And also, as indicated, there's an autoreceptor on the presynaptic membrane. And if you look below, you can see I've drawn in like two distinct kinds of receptor types, right? That neurotransmitters and drugs often encounter postsynaptically. One of them we're going to see, before the neurotransmitter binds in the extracellular space, right? It's going to be closed and have no opening from one side of the membrane to the other. But when the neurotransmitter binds, you can see that little asterisk, it's going to open a hole, right? That's going to allow for ions to pass through. That's going to allow for redistribution of ions across the membrane. Remember that postsynaptic membrane is at rest, right, at -65. So ions are going to move, and it's going to change the polarization, right? So this is a specific class of receptor that we call ionotropic receptors, and we'll talk about those in a moment. And then the other kind we're going to see, there is another protein. So this is the receptor embedded in the membrane. This is the cleft coming, this is the neurotransmitter coming this way, right? I'm inside the cell. And then, on the inside of the cell is, attached to this receptor is another protein. It's called a G protein. And there's many different kinds of G proteins we will see. And when the neurotransmitter binds out here, right, binds, there's a change in shape, like there always is when a chemical binds another chemical. And the result is the release of that G protein inside the cell. So that G protein can now go off and do many things because the neurotransmitter has bound out here. So it's going to cause an intracellular sort of metabolic change. These are called metabotropic receptors. And we're going to see these two types of receptors, two classes of receptors, two families of receptors are very important in both neurotransmission, chemical neurotransmission with neurotransmitters, and also the actions and effects of drugs.